Have you ever wondered how our genetic information is copied? It's a fascinating process, and it's happening right now in every cell of your body. It's called DNA transcription, and it's how the genetic code in our DNA is copied into a complementary RNA molecule. So, how does this process work? Let's break it down into its main steps: initiation, elongation, and termination. The process begins in the nucleus of cells with a molecule called RNA polymerase. This molecule binds to a specific region of the DNA, known as the promoter. This region marks the start of a gene, and it's where the transcription process begins. The RNA polymerase unwinds the DNA double helix, creating a transcription bubble. The strand of DNA that serves as a template for RNA synthesis is called the antisense strand, while the other, the sense strand, remains intact. Next comes the elongation phase. The RNA polymerase synthesizes the RNA molecule by adding nucleotides that are complementary to the DNA template strand. This happens in a specific direction, from the 5' end to the 3' end of the RNA molecule. The base pairing rules are followed, but there's one key difference. In RNA, the nucleotide uracil, U, pairs with adenine, A, on the DNA template strand, instead of thymine, T. With each step, as the RNA polymerase moves along the DNA template, the RNA molecule elongates. Finally, the process ends with termination. The RNA polymerase reaches a termination signal on the DNA template, which causes it to dissociate from the DNA, releasing the newly synthesized RNA transcript. In prokaryotes, these termination signals often involve hairpin structures formed in the mRNA, while in eukaryotes, they may involve specific sequences and protein factors. But that's not the end of the story for eukaryotic cells. The newly synthesized RNA transcripts undergo several modifications before they become mature mRNA molecules ready for translation. These modifications include adding a cap at the 5' end, adding a tail at the 3' end, and removing certain sections called introns through a process known as splicing. Now remember, mRNA isn't the only type of RNA molecule. Transcription also produces transfer RNA, tRNA, and ribosomal RNA, rRNA. The tRNA carries specific amino acids to the ribosome during the process of translation, while the rRNA forms part of the structure of ribosomes where protein synthesis occurs. Transcription is a highly regulated process. Various mechanisms, including transcription factors, enhancers, repressors, and epigenetic modifications, such as DNA methylation and histone acetylation, control the expression of genes in response to different signals and needs. So there you have it. DNA transcription is a complex yet beautifully coordinated process that turns the genetic information in our DNA into functional RNA molecules. These RNA molecules play a crucial role in gene expression, cellular processes, and the development of organisms. It's truly a marvel of nature.